infect the human cells. So following the binding of the RBD to the receptor ACE2, actually the SARS-CoV-2 virus, the membrane structure will change and fuse it together with the target cell or the human cell and lead to the release of the viral nucleic capsid into the cytosol of the infected cells. And the virus will start replicates in our cell and get ready express and explode for the next infection. So during the infection, our immune system, especially the human immune system, is also plays a very important role to help us and protects us to fight against the virus infection. So after receiving the antigen stimulation, the B cell from the native B cell, naive B cell will activate it and they begin to a series of proliferation or differentiation to form a plasma B cell, which can produce the antibodies, and the antibodies can help us to fight a better fight against the virus. So here we show the kinetics and almost based on the structure and function, the antibodies in our body can be divided into the IgM, IgA, IgG, and IgE. And usually the IgE is more related to the allergies, and the IgM, IgA, and IgG plays very important role in our immune system to fight against the different diseases. So the figure here shows the natural infection of the SARS-CoV-2 virus and how the antibody changes during from the very beginning to the long term. So as here we can see at the very beginning after infection around 10 days, the IgM and IgA, the level is increased very fast. So the IgM and IgA can provide the very fast and rapid protection to fight against the virus. However, the half-life of the IgM is very short. So after like 14 days after the infection, the IgM, the level is start to decrease. And for the long-term immunity, the IgG is the most important one. So usually after around 10 days after infection, the IgG started to increase in the circulation system, and we'll get into the peak after around 3 to 4 weeks after infection, and that will keep existed in our human bodies and provide human a long-term immunity. However, for the different diseases, the half-life of the IgG also different. For the SARS-CoV-2 virus, we still not know very clearly, so we have to put a big question mark here. But I think it's very important to continually check the IgG levels changing, especially for the convalescent samples or the people after vaccination. It can help us better prevent the diseases and to also to cure the diseases. So what's how the antibody protect our body, or how the antibody fight against the virus? So based on the function, actually the antibody can be divided to neutralizing antibody or non-neutralizing antibody. The neutralizing antibody persists in the circulatory system, could bind to the pathogen in the bloodstream. Partially of that could block cellular infiltration and replication and protect our from the superinfections or reinfections. Based on the current published papers, the neutralizing antibody to against the SARS-CoV-2 is bind with the RBD protein. So after the neutralizing antibody bind with the RBD, the SARS-CoV-2 virus cannot bind with the ACE2 anymore, so the virus cannot get into human cell anymore. So we can block the virus infection at the very beginning. However, there's also some non-neutralizing antibody that combine with other protein on the virus surface. And for those proteins, even these antibodies can bind the virus, but the antibody binding can still not block the virus binding with the ACE2, and the virus can still get into human cell. And the worstly is there will be some antibodies. After they bind with the virus, the Fc fragment can be recognized by the monocytal or the macrophages Fc receptor, which mediate the antibody mediate the inflammation or the antibody mediate the infection, which make the patient's symptom even more worse and from can be developed to a very severe diseases. So the classic method to analyze the neutralizing antibody is to use the live virus neutralization test or the VNT. The VNT requires handling live wild type or the mutant SARS-CoV-2 virus in a specialized biosafety level 3 or BSL-3 lab, contaminated facility, and in the laboratory intense and time consuming. Usually that will take at least 3 days, but usually that will take 4 to 6 days to finish all the analysis. So for the live virus neutralizing test, because the highly requirements for the facility and the time consuming is not very easy to be achieved in all the lab around the world. And the substitute way is we use the pseudovirus-based virus neutralization test. Actually, for this method, usually we use like the HIV based pseudovirus. We put the spike protein on the pseudovirus, and we also have the cells which can express ACE2 receptor. So because the pseudovirus encodes the luciferase gene, so this the quantity that get into the cell model can be detected based on the luciferase signal. So for the pseudovirus, compared to the live virus one, it's much more safety because it can be done in the BSL-2 lab, but because still based on the cell model, so it's still time consuming compared to the live virus one. The time costing almost the same. It also need 3 to 4 days to finish all the experiments. So now based on the situation, we want to develop a faster and much more easier to use the tool to help us to better understand or better detect the neutralizing antibody. So based on this, we developed the neutralizing antibody ELISA test. So the mechanism is really similar to the pseudovirus or the live virus one. We express the ACE2 protein and coated them on the ELISA plates. And for the reasons, we have the HRP conjugated RBD. And so when we mix the HRP RBD together with the serum samples, if there is some neutralizing antibody that will bind with the RBD. So the RBD will not attach to the place anymore. So after washing, the signal can be checked. If more free RBD bind on the place, the signal will be higher. But if a lot of RBD were neutralized by the antibodies, the signal will be very low. So based on the signal, we can detect the level of the neutralizing antibody in the different samples. So here is a summary of the advantages and disadvantages between these three methods. So for sure, the VNT or the live virus neutralization test is the golden standard for study the neutralizing antibody. But compared to the pseudovirus experiments or the ELISA, it requires the BSL-3 lab, and it needs to operate the live virus. So, and it's very dangerous to be done in many lab or cannot be done there. So, and the duration for the experiments, the live virus analysis and the pseudovirus is quite similar because it's all based on the cell model. So 2 to 4 days is required. And the ELISA test, which can be done in the BSL-2 lab, which can be done in most hospital lab. And because there is no virus involved in this experiment, so it's the most safety experiments compared to the previous two. And for the ELISA, the test will be reduced from 2 to 4 days to 1 to 2 hours. So why the neutralizing antibody detection is so important? I think the most important function or most important objective to detect the neutralizing antibody is for epidemiological investigation. So according to the WHO, it mentioned if their sample is positive or suspicious to be either the positive, neutralizing antibody analysis should be ideally be performed, especially with the now more and more people will get vaccination. Uh, so we want to know after the vaccination how the antibodies already created or not, or how the antibody changes with the time after the vaccination, how the antibody will change. We want to know the kinetics. When we should get the reboost, then we can help the better protection by the vaccines. So here is the